So we're going to cover chapter 13 of your textbook. So this chapter is about the control of microbial growth, and control of the microbial growth, it means killing the microbes and also preventing the growth of the microbe. So growth, as you know, in microbes means like increasing the number of the microorganism, and so control of the microbes, so it's not necessarily means killing them, so it can be the prevention of increasing their numbers too. So this is an interesting picture. So you can see that we can find microbes everywhere, especially on fomites, so that all those items that we use frequently. For example, think about like a doorknobs, toys, towels, and also think about your car. So these are the number of CFUs, so colony forming units of the bacteria that you can find in different parts of the car. And so I thought this is interesting for you, that you can see that we are dealing with microbes everywhere, and so it's very important to know how to control the number of these microorganisms and to prevent the spread of the diseases. So first, let's talk about the biosafety levels. We have 4 different levels of the biosafety labs, or BSL. So you are not going to work with BSL 3 and 4 during this course, but usually the lab that we have is BSL 1 or 2. And as you can imagine, as we get closer to BSL 4, the risk of these pathogens or these microorganisms becomes higher. So BSL-1, pretty much these microbes, they cannot cause any diseases in healthy hosts, and they have very low risk for the technicians and the people that they are working with them. For example, non-pathogenic strains of the E. coli, they are in this group. So BSL-2, as you can imagine, they are usually a little at moderate risk or higher risk than BSL-1. So you should do like the same way that you are treating those BSL-1 pathogens or microorganisms, but you just need to be more aware of them and their like risk. BSL-3, so that as you can imagine, these are very, very dangerous and pathogenic microorganisms. For example, Mycobacterium tuberculosis is in this group, and they can be spread through like a respiratory system. So you have to have special equipments in order to work with these pathogens. BSL-4, as you can imagine, these are very, very high risk pathogens that they can spread easily, especially through the aerosols, and Ebola and Marburg viruses, they are in this group. So, and some of them, they cannot have even any treatment or vaccination. So that's why the people that they are working with them, they have to have some special training to be able to work with these pathogens. Before we talk more about the different types of control of microorganisms on fomites or non-animated items, let's learn about some definitions. The first one is sterilization. And this is not an easy process that you can get it at home. So this is a process that you can destruct all the microbial life, doesn't matter viruses, bacterial, fungi, endospores, enveloped viruses. So it means there is no microbial life exists in that like fomite item. And then we have disinfection. Disinfection means destroying the pathogen, usually on a surface, and you are not gonna destroy all the pathogens or all the microorganisms. There are still gonna be some that they are still there, so but you usually can easily get rid of the vegetative part of the microorganisms, and you can use physical or chemical techniques for getting rid of them pathogens. Then we have sanitization. So sanitization, this is the in decreasing the number of the microorganisms. Again, there are maybe some endospores, some viruses, some bacteria that they can survive. So your main goal is that reducing the number of the microorganism to be able to prevent the spread of the diseases. And sanitization usually happens in the commercial establishments, for example, at the hospital, public bathrooms, and like those like commercial areas. You can use high temperature washing, you can use like a disinfectant and like other techniques in order to get rid of or decrease the number of the pathogens. So then we have some other definitions that this is the control of the microorganisms on living tissues. So you are talking about like, for example, your skin or the wound or inside or on inside your body. So these are like the living tissues. So the first one is antisepsis. So antisepsis means removing the microorganism from living tissue. For example, you use antiseptic in order to, usually chemicals, in order to clean the wounds. You use, for example, iodine or hydrogen peroxide for like getting rid of or removing the microorganisms. And then we have degerming. So this is like means scrubbing and removing the microorganism like physically, and it's not about killing. So it means like you decreasing the number of those pathogenic microorganism that we have. For example, before doing the injection, we rub some alcohol on that area of the skin to make sure that we decrease the number of the pathogen that they are not gonna enter the body through the injection. So these are on living tissues. So what protocol we use in order to prevent the growth or the control of the microorganisms? So it depends on the item. So are you working with some surgical tools, or are you working with like a doorknob or I don't know, a room in the hospital? Or so there are like, depends on the item that you have, you can use different kinds of techniques. So sometimes you need to have a sterilized condition, sometimes you need to have semi-critical condition. For example, disinfecting is enough, you don't need to have a sterilization, and sometimes you just need to make sure it's clean. So there are, depends on the item, we have three different categories. The first one is the critical item. These are the items that they go inside the body and they penetrate inside the tissue or your blood, so they have to be sterilized. So we have to make sure there is no living organisms or life on these items or things that we are dealing with. Then we have semi-critical items. So these are the items that they can be in contact with your mucous membrane or unbroken skin, but they are not going to go inside the body or penetrate inside the body. So you don't need to have a sterilization for them, but they have to have a high level of disinfection. And then we have non-critical items. So these are the items that they don't penetrate the skin and they just need to be clean. So they are just in contact with your body, but they are not gonna go inside the body or be in contact with your mucous membrane. So this is a nice summary table for you guys that you can study the different techniques that we use for the control of microorganisms on fomites and the living tissues. So there are different ways that we can measure the microbial growth. And so these are like different definitions that you need to know. One is cidal. For example, bactericidal. So you can see here. So when you see bactericide or bactericidal on, for example, a chemical, it means it can kill that bacteria, can kill the bacteria. Or viricide or virucidal chemicals or techniques, it means killing or inactivating the viruses. Fungicidal means killing the fungus or fungi. So these are like important definitions to know. And then we have the static. So for example, bacteriostatic. So these are these reagents, they can inhibit the growth of the bacteria, but it doesn't mean that it can kill them. Or fungistatic, so this can just stop or inhibit the growth of the fungus. So now we're going to talk about the different techniques that we have in the control of the microorganisms. The first one is the physical methods of the control, and the most popular and important techniques that we use is the heat. It can change the cell membrane function and structure, and also can cause denaturation of the proteins and enzymes in the cell. So we have two definitions you need to know. One is the thermal death point, which is we call it TDP, and thermal death time, which is TDT. Thermal death point, this is the lowest temp or temperature that we use in order to kill all the microorganisms in a 10 minutes exposure. And the thermal death time, this is the duration that we need in order to kill all that microorganism, kill or prevent their growth at a certain temperature. Okay, let me give you an example for each of them. For example, I have three different temperatures: 60 degrees Celsius, then I have 80 degrees Celsius, then let's do 80, and then I have like 100 degrees Celsius. So at 60 degrees Celsius, 50% of the microorganism will die. And then at 80, for example, 100% will die. And then at 100 degrees Celsius, again 100% will die. So here, which one is the TDP, the lowest temperature that I need to kill all these microorganisms at a certain time or 10 minutes exposure, is the 80 degrees Celsius. So it means I don't need to go beyond that in order to kill the microorganisms. So this is the lowest temperature that we need to kill all the microorganisms in 10 minutes. Then the TDT is opposite, so you're gonna have like the same temperature. Imagine I use 80 degrees Celsius, and then I have like 5 minutes, 7 minutes, or 10 minutes, and 15 minutes. So then at 5 minutes, 50% will die. At 10 minutes, 100% will die, and at 15 minutes again 100% will die. So then the lowest time that I need to be able to kill or prevent the growth of this microorganism gonna be 10 minutes. So we call it thermal death time, or TDT. So many people, they think that boiling can kill all the microorganisms, but that's not the case. For example, they think that if they boil certain items, like for example the baby's bottle, or some surgical tools, or so this is not actually enough for killing and sterilization. So it may kill the vegetative part of the cells and viruses, but still there can be some endospores or envelope viruses, or some macroorganisms that they can stay alive and survive at boiling temperature. And so it's not always considered very useful technique in all the cases. So for the heat, we have 3 different ways to use the heat in order to prevent and control the macroorganisms growth. One is the moist heat. So you're going to use the heat, and then you're going to use like a pressure from the steam or from the evaporated water in order to control the microorganism. Then we have pasteurization. Just remember, in pasteurization, we are not gonna kill all the microorganism. We're gonna reduce the number of the microorganism, but it's not gonna sterilize the for example, that juice or alcoholic drinks or milk. And then we have the dry heat. So this is when you use oven or flame in order to prevent the microorganism or kill the microorganism. So usually in the laboratory and the hospital, we have to use sterilization by heat, and for this technique we're gonna use flaming. So when you guys gonna come to the lab, so you're gonna use like Bunsen burner in order to sterilize your loop, or if you're using needles or anything for culturing, so you're gonna use like the flaming. So this is like a dry heat that you can kill all the microorganisms, that all the living particles that we have, or living cells that we have on that tool or item. And then incineration. So we have some in the lab. So it's like a older like technique that they use. There are some labs that still use it. So this is like a tool that they put the loop inside, and then it has a very high temperature. It can sterilize the loop. And but another important technique that we use in the lab, we use hot air sterilization. This is also dry heat, so we have to use very high temperature, for example 170 degrees Celsius for 2 hours, in order to for example, we use an hot air oven in order to sterilize some of the tools that we need in the lab. And just remember, if you use the dry heat, you have to have a higher temperature, and also you need to have longer period of time in order to do the sterilization. So when you use the moist heat, so is opposite. So you can use lower temperature, and you can use a shorter amount of time to do the sterilization. And for the moist heat, one of the most important thing that we use, we call it autoclaves, that we have it in the lab. We have tabletop autoclave, and we have the industrial versions both in the lab. So you can put like all the tools or solutions that you want to do the sterilization for 15 minutes at 121 degrees Celsius. So these are important data that you need to know. And it has a pressure of 15 p SI. So because the heat has like it's moist, so it has water in it, so it can penetrate easier in that like solution or in that item that we want to do the sterilization. So that's why you need shorter amount of time and you need lower temperature in compare with the dry heat in order to do the sterilization of the material. Then we have the pasteurization. As you can imagine, there is no sterilization in pasteurization. So in pasteurization, we just eliminate the pathogens and we decrease the number of microbial organisms in order to prolong the life of the product. We have three kinds of pasteurization. We have classical, so we use 30 minutes at 63 degrees Celsius. Then we have the high temperature short time pasteurization, so that they usually use it for the milk that we get it from the fridge in the stores. So they use 72 degrees Celsius for 15 seconds. And then we have ultra high temperature, so they use it for the milk that they usually they are not in the fridge, so they are in the boxes in the shelf, so they don't need to be stayed in the cold temperature, and they have a longer shelf life. So they use this UHT, ultra high temperature techniques. So these are different techniques of the pasteurization that we use it in order to kill and decrease the number of the microorganisms. So these are some of the microorganisms that we can, the pathogenic microorganism that we can kill during pasteurization. But just remember that we cannot do a sterilization during pasteurization. Next is using cold in order to control the microbial growth. So usually low temperature cannot kill the bacteria or microbes, so it can slow their growth, but usually we don't use like the freezing in order to kill the microorganism. There are some microorganisms that psychrophiles that they actually love the cold, so they can grow easily in the fridge or in the cold temperature. And usually bacteria, they can become dormant, so they can stay like dormant, and then when they get out of the cold, they continue to grow. For example, the lunch meat, when you put it in the fridge, so it's okay, but as soon as if you take it out and after a couple of maybe 1-2 days, then you're gonna see it's kind of slimy. So the microorganism, they were there when they were in the fridge, but when they get out of the low temperature, so then they start to grow and they start to increase their population. So some bacteria can be preserved actually by freezing, so that's why we use the liquid nitrogen in order to preserve our bacteria or our microorganisms, so we can preserve and keep them for years at minus 70 or 80 degrees Celsius. Usually eukaryotic microbes, they are more sensitive to freezing, but heat is more efficient for killing and controlling the microorganisms. So I'm going to stop here, and then we will continue with part 2, so after a short break for you guys.